Hi everybody, this is going to be the fourth lecture of urinary system, and I ended up going a little shorter on the previous lecture, so we are continuing to talk about filtrate processing in this lecture. And let's go ahead and talk about not really attendance questions. Why does someone with diabetes mellitus, and again that's the diabetes that we tend to think of when we think of diabetes, have increased urine output? What is the result of, that should be ADH on urine output. Let me go ahead and fix that. What is the effect of ADH on urine output? What is the result of ADH on urine osmolarity? And what is the result of ADH on plasma osmolarity? So go ahead and pause your video and try to answer these. And let's see what the correct answers are. Why does someone with diabetes mellitus have increased urine output? Well, this is because the glucose that's elevated in the patient's blood is going to also be elevated in this person's filtrate. And remember, in the PCT where glucose is reabsorbed, typically we reabsorb 100% of that glucose. But in the case of someone with diabetes mellitus, they have excess, so that glucose exceeds transport maximum. It pulls water along with it. It resists water being reabsorbed. We say that it is an osmotic diuretic. So by the time we get to the end of the nephron, there's glucose and there is excess water. And the excess water is what results in excess urine output. These next three actually really tie in with the urinary lab. So if you haven't already watched the videos and read the urinary lab, these will really go hand in hand with that lab. What is the result of ADH on urine output? Well, it's the job of ADH to increase water reabsorption. A little bit at the DCT, but primarily at the collecting duct. And if we're reabsorbing more water, there's going to be less water in the urine. So there will be less urine output in response to ADH activity. What is the result of ADH on urine osmolarity? So you can kind of think of osmolarity as concentration. It's not exactly the same thing. Remember, we discussed this earlier. But ADH, if it's reabsorbing water, it's increasing the concentration, so urine osmolarity increases as a result of ADH activity. And what is the result of ADH on plasma osmolarity? Well, we're pulling water out of the urine, or out of the filtrate before it becomes urine. Where does that water go? Ultimately, it goes back into the blood, into the plasma. So ADH decreases plasma osmolarity. All right, so let's move back to our lecture. So last lecture we talked about a broad overview of reabsorption, and then we started talking a little bit more specifically about reabsorption in the proximal convoluted tubule. Today we're going to give a broad overview of secretion, and then we're going to look at reabsorption and secretion throughout the rest of the nephron. So tubular secretion really is like reabsorption in reverse. Reabsorption was taking substances from the filtrate and putting it back into the blood. Secretion is taking something from the blood that was not filtered and placing it into the filtrate. So things like drugs, certain metabolites, products that are produced through metabolism, and other molecules. These things, drugs, metabolites, and other molecules, tend to be bound to plasma proteins. Well, if something's bound to a protein in the plasma, remember, proteins are not filtered. So these substances also will not be filtered. If our body wants to get rid of them, then they have to be secreted somewhere later along the nephron. And nitrogenous wastes. Nitrogenous wastes are really byproducts of protein metabolism. So when we're digesting proteins and producing amino acids, we also produce nitrogenous wastes like urea and uric acid. And now these nitrogenous wastes are a little bit different from the drugs, metabolites, and other molecules because nitrogenous wastes, they actually are filtered. But for various reasons, they are reabsorbed early in the PCT, but then they are almost immediately secreted back into the filtrate along the PCT and later in the nephron. So here's an image from your textbook on page 908, and this kind of sums up what's secreted and what's reabsorbed along the nephron. The things that are in blue are things that are being reabsorbed. The things that are in pink are being secreted. So here's our generalized nephron, here's our PCT, and here's all the stuff we talked about yesterday or last class with the things that are being reabsorbed. And then here are some things that are being secreted in the PCT. Here's that urea and uric acid, which was just reabsorbed, and then it is immediately secreted again. Now we're going to look at along the rest of the nephron throughout today's lecture and look at things that are being reabsorbed and secreted. So after we leave the PCT, we're going to go through this hairpin-shaped area, this U-shaped area called the loop of Hindley or the nephron loop. Now, as we saw back when we were doing cardiovascular, biology is trying to get away from naming parts of the body after people. So loop of Hindley is an older term. Nephron loop is really what it's being named now. But really, loop of Hindley was around for so long, people got used to using it. So when you get into your programs, if you talk about the kidney, you're still probably going to hear something like loop of Hindley instead of nephron loop. But both of those terms work. And the loop of Hindley, or the nephron loop, has two separate sides. One side, immediately following the PCT, goes downward. This is the descending limb of the loop of Hindley. Then we go around this turn and we come back up. This is the ascending limb of the loop of Hindley. Now, when we look at the loop of Hindley, there's also this area called the thick segment and thin segment. On the descending side, there's not really too much difference. They kind of do the same thing. But on the ascending side, I don't like the way that your book illustrates it because it looks like the thin segment is way down here and that's as far as it goes, when in reality the thin segment goes quite a ways up. And then we get the thick segment. And that's going to be important for something we talk about here in a couple of slides. So keep in mind, thin segment on the ascending side is much longer than your book makes it look. So first let's look at the descending limb of the loop of Hindley. And this side is permeable to water. And water is being reabsorbed the entire descending side of the loop of Hindley. But when we go up the ascending side, this side is permeable to solutes, but nothing else. And the thin segment is reabsorbing sodium and chloride, and they're being reabsorbed passively, just like we saw in the PCT. Sodium and chloride being reabsorbed, no energy is being required. We'll see the mechanism for that in our next lecture. But urea is also being secreted, and it's being secreted down here towards the bottom, really on both sides. It shows it over here on the descending limb, but it's happening on both sides. Urea is being secreted into the filtrate. But now on the thick segment of the ascending limb, we're still reabsorbing sodium and we're still reabsorbing chloride, but we're reabsorbing potassium also. And now we're into an area that's active. So this thick segment of the ascending limb is active reabsorption. It's using ATP. And it's using ATP to power a co-transport mechanism, and that ATP causes us to reabsorb sodium, potassium, and 2 chlorides. Some of the potassium is then going to go right back into the filtrate. On the basal side of that ascending limb, so this is the side closest to the blood, the sodium-potassium pump is moving sodium into the interstitium so that it can pass back into the blood. Chloride, with its negative charge, is following the sodium. And then potassium is diffusing out. So all that sodium and chloride and potassium is still going into the interstitium and ultimately into the blood. We'll see what actually happens when it's in the interstitium next class. So up to this point, the PCT, the descending and the ascending loops of Hindley, those are constantly at work and they're always doing the same job. It doesn't matter what else is going on, they are always doing the same job. But from this point forward, the distal convoluted tubule and the collecting duct, they change their jobs based on the state of the body. So this is where things like hormones are mostly going to work. Now, hormones will work elsewhere, and they may increase or decrease certain activities, but the distal convoluted tubule and the collecting duct are where we see most hormone activity. So first, let's look at when aldosterone is present. We've already talked about what causes aldosterone to be present, but let's look at what happens when aldosterone is present. Sodium reabsorption increases. In the DCT primarily, but also a little bit in the thick segment of the ascending loop of Hindley, and a little bit in the collecting duct, but primarily it's working in the DCT. And it's causing sodium reabsorption, chloride comes with it electrically, and it's causing potassium secretion. How does it cause sodium reabsorption and potassium secretion? We saw that last class or 2 classes ago. It tells the cells in this part of the tubule, in the DCT, and a little bit over here and a little bit over here, but it tells the cells here in the DCT to make sodium-potassium pumps. We reabsorb more sodium, we secrete more potassium in response to aldosterone. Atrial natriuretic peptide, ANP. We saw that way back in cardiovascular. Elsewhere, in addition to the tubules, elsewhere it dilates the afferent arteriole. If we dilate the afferent arteriole, that's not right. If we dilate the afferent arteriole, we would increase NFP and GFR. So that's a typo on here. But ANP lowers ADH, it lowers aldosterone, it lowers renin levels. And it has an effect on the vasorecta that we'll see in our next class. ANP also interferes with the activities of ADH and aldosterone that may already be working. So it prevents ADH from doing its job in the collecting duct and the DCT. It prevents aldosterone from doing its job in its 3 areas. So ANP is having the opposite effects of what ADH and aldosterone do together. So what does ADH do? We've already said it increases the number of aquaporins, specifically aquaporin type 2, which increases water reabsorption. It also increases urea reabsorption in the collecting duct. So over here in the collecting duct, we're always reabsorbing a little bit of urea, but ADH causes it to reabsorb more urea. Why would we want to reabsorb urea? Well, let's think about that. Urea is a solute. If we take urea out of the filtrate and put it into the interstitium and then ultimately into the bloodstream, what do solutes do? Solutes suck. It's going to pull more water along with it. So by reabsorbing more urea, we're going to reabsorb more water. Now, ADH functions in the DCT and the collecting duct, but it plays a bigger role in the collecting duct, just the opposite of aldosterone. Aldosterone works more in the distal convoluted tubule. ADH works more in the collecting duct. But let's tie those two together as a thinking cap question. We've kind of touched on this already. Why do ADH and aldosterone always travel as a team? Think about that and then see if you can come up with an answer before we go over it. Well, ADH, it increases aquaporins so water can pass through. But water doesn't really have much reason to pass through on its own. Aldosterone causes increased sodium reabsorption. Well, we can increase sodium reabsorption, but if that's all that we do, water can't come along with it. We have to have ADH to provide the pathway, those aquaporins, and we have to have aldosterone provide the pulling force for that water in the form of sodium. So aldosterone provides the pulling force, ADH provides the pathway, and the ultimate result is increased water reabsorption. They have to work together because neither one can do anything on its own. Continuing with the distal convoluted tubule and the collecting duct, in response to PTH, the parathyroid hormone. PTH, we talked about a little bit in Bio 137, but here in the kidneys, PTH causes increased reabsorption of calcium in the distal convoluted tubule. It really works throughout most of the nephron, but most of its effects are in the distal convoluted tubule. So we reabsorb more calcium in response to parathyroid hormone. It also stimulates the activation of vitamin D. Remember we said that the kidneys activate vitamin D? Well, they do that in response to parathyroid hormone. And when we get to endocrine, we'll see vitamin D is actually needed for our body to absorb calcium from our diet. If we eat something or drink something that's got calcium in it, it would normally just pass straight through our digestive system and leave in our feces. But PTH tells our small intestines to reabsorb more calcium or to absorb calcium from our diet. PTH is all about increasing calcium levels of our body. One way it does that is increasing calcium in the DCT. Another way it does that is by activating vitamin D. pH maintenance. Our kidneys and our lungs work together to maintain pH. We saw back in the lungs that we breathe to control our pH levels and to control our CO2, which indirectly controls our pH levels. Well, our kidneys are the other organs that are really responsible for controlling our body's pH. And they do that by altering the levels of bicarbonate and the levels of H+ in our blood. When we are too alkaline or when we are too acidic, then they will either reabsorb or secrete more H+ and more bicarbonate, depending on if we want to increase or decrease our body's pH. But that's really going to be the focus of our next chapter when we talk about pH. We'll come back to that, but just know that if we have too much or too little H+, if we have too much or too little bicarb, our kidneys will secrete or reabsorb more pH depending on what it needs to do. And It does that all along the nephron, but primarily here in the DCT and the CD. So that's where we will stop this lecture. Our final lecture over kidneys will be our next lecture, and we're going to talk about how does our body maximize water reabsorption. We're going to talk about the loop of Hindley in a lot more detail, and then we're going to talk about a couple of disorders. All right, so take care, and I will talk to you in just a few days.